My name is Sébastien Degault. I'm working at CYTOO as a project R&D project manager.
Okay. And David, can you just say how that came through on your end?
That sounds fine, thank you.
Okay, thanks. Good. And so I'm working at CYTOO for now 3 years, and I'm happy to be here to present you the latest developments on the 2D Plus technology, which is basically what we'll be talking today a lot is the CYTOO Maps, which is an enabling tool for phenotypic screening. Next slide, please. So the main objective of this meeting will be first to recapitulate what is 2D Plus for the one of you who doesn't know the technology yet, and we will, through concrete examples, try to demonstrate the benefits of this technology in cell-based assay. And then we will introduce and talk a lot about the CYTOO Maps, which we believe has the potential to become a gold standard in phenotypic characterization of drug effects. So I will describe what it is, and I will give you an example, actually, that we did on the subset of drugs from the first evaluation of the DSEC consortium. And then, of course, we can discuss after that to get your feedback on what you think about the CYTOO Maps. Next slide, please. So this is a summary of my presentation today. So I will just also in 3 slides describe with CYTOO and what is 2D Plus technology. Then we'll do a summary of the first CYTOO technology evaluation, quickly to introduce then the CYTOO Maps as the work we have done on the false negative compound, a subset of the library that we retested with the CYTOO Maps. And then we'll have the open discussion. So with CYTOO, yes, next slide. So CYTOO was created in 2008. And the company is based on the discovery of, from the Institut Curie and the CEA, which is micropatterning, and I will describe what is micropatterning a little bit later. From 2008, we grow a lot. Actually, now we are up to 35 people working at CYTOO, and this includes 10 PhDs. The headquarters is located in Grenoble, and there we have a team of 28 people that are either part of the R&D team and developing applications on micropattern in our lab, or also people that are in the clean room manufacturing facilities producing our products. Since its creation, we have now a subsidiary also in US since 2009. We are making revenue, and we are well financed by venture capital. Can you go to the next slide? So these are the list of investors, but we have also, in addition to these investments, 3 large R&D projects that are financed either by the French government or the European Union. And we have a strong intellectual properties status with 3 licenses from the Institut Curie, Harvard University, and CEA, which cover some part of the micropatterning technique. And as CYTOO is growing, we have now up to 30 publications from our customers. We get awarded for that growing and that high profile of, for the CYTOO Society by different awards from the French government.
And that is 30 publications last month, not just 30 publications.
I think it's last month, okay. Last month. Which is better. Next slide, please. And so our customers, I think we have now more than 300 customers worldwide.
Okay.
Yep.
30 last month. I thought, isn't that what Chloe said? Is it 30 total? Or
I think it's 30 total.
Okay, 30
total.
Last month. Okay.
But it was last month. But
yes, okay. And they range mostly from
academic groups.
It's usually mostly academic groups, mostly scientific papers, questions. And we're pleased with the fact that it's overall very high quality. We have PNAS, Nature, full articles in Nature, Nature Methods, Nature, Cell Biology, we have the DSEC and so on. So very high level papers in average. It means that many of the key opinion leaders have already found the technology.
So as I was saying, we have now over 300 customers worldwide, and mostly in the US and Europe. Our customers are large pharma company, biotech company, but also academic center of high level like MIT, Harvard, Institut Curie in France. So next slide. So now I will describe a little bit what is 2D plus technology, and as I said, it's based on micropatterning, and this technology restores some physiological cues that are found in tissue for the cells. Can you go to the next slide? So in vivo, when you look at cells, and especially epithelial cells, you can see that in the intestine tissue of a mouse, like here, the cells are very well organized. They are packed together, they are aligned, they have almost the same volume, and they cannot move in the tissue. They are blocked from each other. And they also exhibit a highly reproducible internal organization. As you can see here, the dots, the green dots above the nuclei corresponds actually to the centrosome, so the cells are all polarized in the same way, and they have very little motion. So this internal architecture comes from the different cues that each individual cells receive from the microenvironment. Next slide. So this microenvironment provides the geometry of the cells, then the contact with the neighboring cells, and also the contact with the extracellular matrix below the cells gives some cues that helps the cells to organize. This composition can vary in function of the tissue, of course. And then also the basal membrane is not completely stiff. There is some softness, so it can it depends also on the tissue, but it can move. And for the epitheliums to grow and keep its function, you need some dynamics. Cells need to divide, and to divide correctly to integrate the daughter cells into the epithelium, and so that it doesn't disturb the overall architecture. So based on all these cues from the microenvironment, cells are very well polarized, and they eventually grow a primary cilia, which cilia actually is present on all cells in our organism. But then when you grow cells on 2D cell culture, next please, then cells are growing on a complete uniform substrate, which is infinite, so there is no restriction, so cells can move, and the substrate is stiff. So when you look at cells in this environment, next slide, I think I lost my movie. Okay, so on the right side, you would have seen cells that are on the 2D culture, and they have variable shape. Well, you can see that on the left, variable size, but they also move around all the time when they are not at confluence. So when cells are in adherence in the standard condition for cell culture, they exhibit high morphological variability and isotropic motion. So next slide. And what that micropatterning is try to restore some of the physiological cues found in the microenvironment, and especially we can act on the geometry of the cells, on the composition of the extracellular matrix, and all of that contributes to restore some of the physiological cues. Can you go to the next slide, please? Do you have any questions on that? Okay. So what we are doing at CYTOO, as we are developing micropatterns for high content cell-based assay, the basic of our product is glass slides that we cover with a cytophobic surface. So the cytophobic surface here in dark is just a cell repellent. Cells cannot adhere. And next. Then on this cytophobic surface, we, okay, yes, I think there is some problem with the animation. Next. Okay, well, it's what we get. Anyway, so on this cytophobic surface, then the micropatterns are created by photolithography. Basically, with UV light, you can destroy the cytophobic surface at specific space and create these micropatterns. Then next. Next. Okay, so here, and then you can, in these holes created in the cytophobic surface, coat with the adhesion molecules you want. So it could be fibronectin, laminine, collagen, whatever you want. And then when you seed the cells on top of this micropattern, as you can see here, cells will go on top of the adhesive region and only there, and then they will take the shape of the micropattern. So here you have a large fibronectin band where you can see that cells have various size and morphology. It just looks like the standard cell culture. And then on the left, you have a disc micropattern. On the right, a crossbow micropattern. And then you can see that the cells adopt exactly the shape of the micropattern. Next. And these are normalized cells. Oh, next. And we distinguish two different types of micropattern, the fully adhesive one on the left and the non-fully adhesive one on the right, which is the cross. Next. Next. I don't know. You're going backward. Oh, this is okay. Next. So when cells are on a disc micropattern, which is basically the same as the uniform surface, except that you restrict the adhesion area proposed to the cells to a small disc, then cells adhere on top of the micropattern, and you have a reproducible geometry, you avoid constant motility, and you keep the cells in a stable state of contraction. So you restore some of the physiological cues, and upon finding the right size for the cells, you can even grow a primary cilia in this case. Next. If we go to the crossbow micropattern, it's just as a disc, next, in which you remove part of the adhesive surface, and you have a crossbow micropattern, which disappears again. I don't know why. Here. Okay, and in addition to the previous parameters, reproducible geometry, avoid constant motility, and keep the cell in a stable state of contraction, then you can add up a new parameter to the cells, which is a reproducible internal organization, and this is presented on the right. When we looked at over 75 cells, this is in order the localization of the centrosome, the Golgi, and then the nuclei, and you can see that the orientation of the cells based on the nuclear centrosome axis is almost always the same, and is going up, such as in tissue. So basically, by using this technology, you can restore some of the physiological cues that are found in tissue. And next slide. And this actually, 2D plus is basically then a guided cell behavior where we can engineer the cell adhesion proposed to the cells through the micropatterns, and this provides a similar blueprint and interior design. And when you do, when you look at the cells in the classical cell culture conditions, they have random cell adhesion and behavior, and you have assay variability. When then you use and you shift to 2D plus micropattern cells, then you have reproducible cell architecture, and this tends to reduce the assay variability. Next slide. And then this technology, one can apply it to a large range of assays, and these are some assays listed here, such as protein localization, internalization, and trafficking. You can study also mitochondria networks or celiogenetics. These assays are mostly based on single cell analysis, but we are now also developing assays based on micropatterning for multiple cells, such as myogenesis, neuronal networks, and even 3D cultures. And there are ways to normalize these 3D cultures on micropatterns. And of course, all these applications can do with whatever cell lines you want to use, primary cells or even stem cells. Do you have questions about the technology? Okay, so next slide. So then I will just briefly go through the first CYTOO technology evaluation through a quick summary. Next slide. So the objective of the first evaluation was to evaluate the benefits of the 2D plus technology for its ability to improve or assist the prediction of adverse effects of drugs. And this project was built to compare two conditions, micropattern cells versus non-micropattern cells. And to do this comparison for this type of assay, we used the Cellomics platform with their pre-existing analysis methods and we looked for actin distribution, cell morphology, and nucleic morphology. And we were supposed to do a two-step evaluation with a full set of drugs on inner cells, then a subset of drugs on FG2 cells. Next slide. So the test compounds library was composed of 50 drugs, 44 different drugs and 6 duplicates that were randomly distributed on the plate and blinded when we got them. Then on each plate we have also controls. These 50 drugs were chosen in 5 different categories. The class 1 was molecules that impact actin filaments. The class 2 impacts microtubules. The class 3, classic toxicants. The class 4, false negative, which corresponds to drugs that went too far in their development, but has some toxicity that were detected later. And then the class 5 was some negatives and other. Next slide. And our summary after all this experience was that we get basically an overall similar detection level of drugs in the different classes in both pattern and non-pattern condition. And you can see here that 31 drugs were detected for the non-pattern condition and 33 with the pattern condition. The only difference where there was some benefits for the micropattern condition is that we have more parameters scoring with the celomics analysis on the pattern condition. So in certain way, it can help the prediction of drug effects. And interestingly, we used also, in parallel to the celomics method, a method analysis called the CYTOO mode, which takes benefits of the micropatterning of the cells and the L shape. And this method just looks at the curvature of the cells. And using this method, we were able to pick up 4 drugs, and that were not detected with the celomics analysis, and they were all in the false negative class. So this was an interesting result, suggesting that if you are using analysis method that exploits a little bit more the benefits of the micropattern technology, then you might improve the prediction of drug effects. Next slide. So overall, the work done in this first evaluation allows the detection of drug effects on both pattern and non-pattern conditions using the celomics analysis. We were able to detect acute toxicity in both cases with similar sensitivity, and the only benefit for the micropattern in this example was a higher number of parameters for the detection of drugs. And then the CYTOO mode picked up a few false negatives. What didn't work as initially planned was the compound plates that were partially compromised due to cross-contamination and precipitation, basically. The data we got was based only on 1 experiment, and we couldn't do the FG2 at that time because the micropatterning protocol was not complete during the project, and now we are able to do it. So based on the data, the evaluation was not fully complete, so we did want to show that the CYTOO technology and by exploiting better the features of the micropattern, we could added value to the predictions of toxicity using micropattern. So that's why we decided to exploit the CYTOO map technology that I will describe now to try to further characterize some compounds. Next slide. Thank you. So I will just give you the introduction of what is CYTOO maps, and then I will show you some results we obtained on the false negative compounds. Do you have a question up to here? Okay. So the CYTOO maps. So CYTOO maps is a technology that helps to measure the difference between fluorescent signals and measure the difference between compartments that are difficult to qualify, such as lysosomes, vesicles, but it can be applied to almost any compartment or any cellular marker in the cell. And it works like that. You need to use micropatterns to do that. You seed your cells on top of micropatterns, they normalize. Then you visualize your compartments through any cellular markers. You do your acquisitions, and then you can align tens of cells. Actually, 50 to 100 cells is enough. And you can align them with the micropattern, using the micropattern. And from there, you get dots representing all your vesicles and all your staining in the cell. And that is used through a mathematical analysis to create a density map, which is presented on the left. And next step, so once you created this density map, then the discovery from Bruno Good Labs and Tam Jong and Christine Schauer, which they patented this method, they now have a method to compare these density maps together and define when there is difference or not. So in this example, next, they were interesting to look at, is there a morphology difference on the Golgi apparatus plus some vesicular bodies around it between the nocodazole and cytokalatin D. So on the top, you have just one image of the staining, and you can see that there is a clear difference between control and nocodazole. But if you compare by eye the control and the cytokalatin D, the difference are less obvious. So what does CYTOO maps is able to compare the density maps from the controls to the treated condition. Next. And so you can compare both the control to the nocodazole and the control to the cytokalatin D. And what does the calculation is just create a p-value to know whether this is significantly different. And in this case, it was. But then you can also compare both the nocodazole and the cytokalatin D together to know whether they are also significantly different. Next. And next. Okay, so like that you can cross-compare all your test samples, and it was shown in a publication published by Christine Schauer and Nature Methods that actually you can detect effects with very low cell number. About 20 cells, 20 to 50 cells is enough to create these density maps, and adding up more cells doesn't change the density map per se. And here on the bottom panel, they show also that you can find a difference between two density maps on micropattern condition, but you will never be able to see this difference for cytokalatin D on non-pattern condition. So you can detect effects with few cells, and on this type of staining for the Golgi apparatus, the variability that you have into your cells doesn't allow you to define there is a difference when you treat with some drugs. Next slide. So this technology of CYTOO maps that's the types of things we would like to do with the CYTOO maps. We would like to take an unknown drugs and compare, make the profiles upon treatment for an organelle, such as the Golgi apparatus. And then we would like to compare these CYTOO maps to the reference bank compounds. So here I presented the reference bank compounds with 4 compounds only, and 6 dilution of drugs. And then the goal will be to test your drugs. Then next. One. Okay, and then basically, so you bid your map for one drug at one concentration, and then you will scan the whole reference bank library to try to compare these maps to the maps from the reference bank. And from there, you should be able to detect that the map on the left for the unknown drug just fits this map for the cytokalatin D. And by increasing the number of drugs from the reference bank and the number of cellular markers, we hope to do profiling of well, we hope we will be able to do profiling of compounds, and we know that this type of profiling will help to phenotypically characterize the effect of unknown drugs. Okay, next. So this technology is compatible basically with different magnification from 10x to 100x. You can use also different types of micropatterns. You can analyze these density maps in 2D, in 3D, or even in time-lapse experiments. Next. Then you can do all fluorescent markers you can imagine. So these are 4 examples, and I will give you 2 or 3 more later. And this is fully compatible with high-throughput analysis. Next. And for example, this is a presentation that was part of the that was an Innovation Award nominee at SLIS this year. So this work was done by Christine Schauer and Taryn Blanc in the team of Bruno Good at the Institut Curie, and they used the organelle map technology to characterize 200 kinase inhibitors, looking at two compartments, Golgi and lysosomes. As you can see here, they could characterize the effect of the kinase inhibitor easily. On the right, you have a difference from the controls for the CDK or the PKG cell line. So we know this strategy is good to characterize inhibitor, and we hope to also use siRNA library screens to better identify the real target and the effect, and to avoid having kinase inhibitors that target several pathways at the same time to better characterize the maps, the change of maps due to the inhibition of only one gene. We hope to combine that with the siRNA. Next. So overall, we hope to apply the CYTOO maps to do phenotypic profiling. CYTOO maps combines a visual and user-friendly interface with a robust analysis for unbiased quantitative cell biology. It can be used with any cellular compartment or substructure which has a specific marker. It's highly sensitive. It detects very small effects that couldn't even be observed on standard conditions, and you have statistically significant results on very low amounts of cells. Next. Do you have a question on the CYTOO map part?
So, like when you're looking at these different factors, you've got these maps, but are you somehow quantitating these different particles you're looking at? Is the map quantitated, or is it just a pattern?
So you do the density map, which is basically, let's say you have vesicles in your cells, you take 50 cells, you pile up all the signals from all the cells, and then you look at the density maps, which is basically where the signal is over these 50 cells. And then the statistical methods used compare the density maps from one condition to the other. And that's where the mathematics are. And this tells you whether it's significantly different or not.
How reproducible is it with the same compounds from study to study of whatever result?
I can show you maybe on the maps later, right? On the plate. So you will see from well to well the density maps different, but basically it's reproducible over time between experiments. And that's basically what we did here. The experiments on the false negative. Well, let's go to the next slide. What we did is, because the first DSEC evaluation was kind of incomplete, and we had this development on the CYTOO maps, we wanted to apply a dedicated analysis that exploits the benefits of 2D+ technology, and not only with the morphology, such as the mode analysis that I presented, but here with the CYTOO maps you can look into the cells and the benefits from the normalization of the internal organization. And so what we did here is to take 4 compartments, actin, lysosomes, Golgi, mitochondria, and we took the false negative from the preliminary DSEC evaluation, and we built CYTOO maps, and we looked just whether we would be able to detect effects on the CYTOO maps. And our experiments was done with triplicate points, triplicate experiments done at different times, and we got pretty good results in reproducibility. And we are still analyzing all these data because we collected the last data points 2 weeks ago. So I can not give you numbers right now, but I can show you the maps for certain plates, and you will see triplicate points on the same place. Can you go to the next slide? So this is typically the type of experiment we were doing. This is what we call the control plate, and so there were 4 drugs, 6 dilutions, 110, so going from 10 micromolar to 0.1 nanomolar, and each time on each plate we have triplicate points. And at the bottom you have the DMSO control, 12 DMSO control, and for each plate we have ibuprofen, which was negative control, nocodazole, which targets microtubules, cytokalazinie, which targets actin, and CCP, which targets mitochondria, and these were present on all the plates. So that's where we looked at reproducibility of the signals over the plates in one experiment and across experiments. And here you can look at the maps for let's say, the nocodazole at the highest concentration, and you can see that the maps are pretty reproducible. And if you look at the bottom, you have also the nocodazole. The concentration down there is 1 micromolar, which corresponds to the 8th well, the second well in the nocodazole. And these maps are also pretty consistent. So when you do, can you click next? Yeah. So what we did actually is to simplify the analysis, because you can do the comparison across all the wells if you want. We just pile up the 3 maps together, and then we compare the nocodazole at the bottom to the dose response, and it was not significantly different from the right concentration, but then it was significantly different from the lower concentration. So you can really, yeah, that would be helpful. I can see. Yeah. So this nocodazole is actually the same concentration than here. And this is the CCCP, which is right here. So you can see that this looks like exactly like that, and we got that all over the place. And when Christine Schauer did the first experiment, actually she reproduced results from Manuel Thierry that did exactly the same experiment 5 years ago. So, and she was, this is in her paper actually. This is really reproducible technique for that. Yeah, so next slide. So, and the way we identified it for the false negative, it's basically drug at a given concentration is considered as a hit when it is significantly different from the control in at least 2 out of 3 experiments. Okay, next. So this is just an example on the controls over 6 different drugs. This is the types of results we get. So CCCP, these are the maps for the Golgi apparatus. For CCCP and ibuprofen, we don't have any effect actually, or the effect is higher than 10 micromolar. And for all these drugs, which are targeting for these 2, the microtubule, and for these 2, the actin cytoskeleton, we can detect an effect at different concentration depending on the drug. For cytokinin D, it's up to 10 nanomolar, which is here. Y27632, it's over here, and for the nocodazole, it's up to here, and for the Taxol, it's up to the lowest concentration. So we're able to detect effect with the Taxol up to the lowest concentration. And next slide. And when you look again at the Taxol, but over the 4 different stainings, then for these drugs, we were able to detect effects at the lowest concentration on all organelles. And these are the control maps. And you can see that there is a difference at each concentration. So next slide. So what we got for now the false negative compounds, it's actually if the true positive have a large cellular effect, such as here for nocodazole and cytokinin D, for all the false negative compounds, the effect was way smaller, but significant on some of the cellular markers. And next slide, please. So this table summarizes the effects we were able to catch up with the CYTOO technology. So the controls are here, and as you can see, Taxol, nocodazole, cytokinin D, and Y27632 actually have an effect on all organelles. CCCP, which targets specifically mitochondria, has an effect on mitochondria, but we could detect also at the highest concentration an effect on lysosomes. And ibuprofen, which was our negative control, didn't have an effect on 3 organelles, but did have some effect at 10 micromolar on actin. But we saw some publication indicating that indeed ibuprofen can interact with actin without completely disrupting its function. It can modify probably its distribution in the cell at some level. Then when we look at the false negative compounds, 2 of them affected 3 organelles out of 4, which is captopril and lidocaine. A lot of them were affecting mitochondria, such as methotrexate, dapsone, busulfan, and lomefloxacin. And some of them affected other organelles, such as actin and lysosomes. And 2 of the compounds didn't affect, actually, or 3 of them, no, 2 of them, didn't touch any, we could detect any effect for these two compounds on any of the cellular marker testing. So next. So based on CYTOO maps, we demonstrated that we were able to detect effects of the compounds on different organelles, and this was done at 4 hours. The positive control showed a large response, as expected, and the negative control, such as ibuprofen, didn't show any change in organelle map distribution. We are clear here that there is a limit in our classification because it is difficult to associate an effect on the modification of the morphology of any compartment to real toxicity. But we hope that by building a reference compound library, this method has the potential to become a gold standard in the quantitative analysis for phenotypic profiling. And I think I'll stop here for now, and I can discuss a little bit more on the future of CYTOO maps. François or Lisa, do you want to?
We have more slides here on the light. So we can have a light for the rest of the slides anyway. Do a couple of slides then. That's the last one. I don't understand your statement on the bottom. You're saying false negative test compounds have a smaller but significant effect, but then for lidocaine you report out 10 nanomolar.
Yeah.
How do you define
smaller effect? So it's either different or not. That's what you would say. But the p-value, like, so what we get from the comparison of the CYTOO map is the p-value. And when you compare the control to the nocodazole for like the highest concentration, you will get a p-value that is like 10 minus 200, which is like way significant. For the captopril or the lidocaine, then the difference was based on 10 minus 3 or 4. It's very, it's different, but it's not as much as the other one. So that's why we are seeing smaller effects. It's first because we cannot see by eyes, and when we see by eyes, it's like 10 minus 50 at least. But there is still a statistical difference between the maps. So that's what we call smaller effects.
Sorry, we couldn't hear you in the audio on, so do you want to repeat?
Oh, I had asked how Sebastian would define smaller but significant. And I was referring to lidocaine because in the table it has a potency of 10 nanomolar. And so I wanted to understand how he came up with that value if the effect was so low.
Yeah, small is just based by the p-value that is given by the test, which is smaller in the case of lidocaine compared to nocodazole. That's what we call smaller.
It doesn't have anything to do with the concentration at which it happens, just the magnitude of
Yeah.
the
Let's talk about membrane potential because that might be the only thing I can understand, right? So you're telling me that then you only have like a compound will only change membrane potential by 20% instead of a whole drop of membrane potential at the high concentration. How would you define smaller for membrane potential, for example?
It's just looking at, we're just looking at a heat map. So essentially it's just the distribution in the heat map changes only enough to show a very small difference, and it's just p-value. So not necessarily percent.
Yeah.
My question is, specifically, what the total intensity of certain stimulants will be taken into consideration?
Sorry, can you speak up a little bit?
I was just asking, the map typically you focus on distribution, right?
Yes.
But I would imagine if your total change, that somehow will change the distribution.
Yeah. So, what?
I'm not sure I understand the question.
Total intensity.
Total
intensity. Yeah.
Because this is focused, the map seems focused on distribution,
Yeah.
not so much on, the total up or down or something, right? Because this is kind of a different angle. So I'm just wondering whether doing the analysis at some point, the total intensity was taken into consideration.
For this analysis, I think it was part of it, but not significant. So we are now developing actually a new analysis where we will put more weight on the intensity because, of course, you can go from a high signal to a low signal, you will still get a density map. But then there is a difference that you don't take into account, which is the intensity and the variation of this. And
Yeah.
So that's something we will include in the future, and I think Mathieu is working on that.
Yeah. It seems to me that the question relates to one of the steps of processing the image where you break the structures in dots. Okay. And you create a cloud of dots. And then you adopt the cloud from different sounds. Based on the probability density map.
Yeah.
And you compare clouds of dots with clouds of dots.
Sorry.
The last version that we are working on, each of the dots has a weight. And the weight is
intensity.
Yeah.
Oh, I see.
But it's not ready yet.
It's not ready yet, but that's an important point.
Yeah, because I imagine, again, like, because the intensity change, the distribution, you will have a kind of different radar.
Yeah, exactly. So that's what we are integrating into
All right,
the
but for
alliance
now,
right now.
I guess the current
For now, for the current
intensity
technology,
itself is not part of the analysis.
no. But we will wait that.
The other question I have, which I think we talked about it last year, about the dosing time. So for now, still no longer than 4 hours. Is that still true?
We do 4 hours for now.
Just want to double check.
If we have low serums condition, we could go for a little bit longer, like 24 hours if we like. But right now, we keep the 4 hours. We kept the 4 hour treatment.
All right.
That means the cells are dead then after 24 hours?
No,
Sorry.
they just
Yell to me.
We're trying to be polite, John.
yeah. The question was, does the cell die after 4 hours or 5 hours or 6 hours? They don't die, but they divide when they are in high serum. And when they divide, there are 2 cells on one pattern, so the normalization is gone. So that's why we are not, right now, when you have serum in the media, you are restricted to the time of cell division, which is basically dependent on the cell line, but 16 to 20 hours. But if you have low serum, then cells don't divide, so you can extend this window.
So I have to just make a point, which is that we also develop network patterns so that cells divide, can invade new patterns and normalize again. So as to lift the limit of the force in our system. And this is a technology that was starting to test. We have several partners in Europe that are starting to use that. And you get individual normalized cells that are division. And we have a flyer of that.
Yes. Yeah.
I have a question around, I understand it's a nice idea of patterning and deriving parameters from there, but going back to the question from membrane potential as well as just regarding fluorescence intensity changes, aren't you kind of leveling the playing field a little bit for CCCP, especially where you would have definitely a huge drop in membrane potential, but that's not being captured in the assay.
That's true.
So you're kind of saying 10 micromole, I mean, it's not good to do of selling, you're saying the effect is on both Golgi and mitochondria, which may not be true if you were to look at just the fluorescence intensity parameters.
Yeah, that's something we will add up to the study. Lisa, you want to comment on that?
Okay, you're doing okay. I mean, that is, we have to do the intensity.
The intensity, yeah.
And I agree with you that that's an important feature.
Yeah.
And then, relying on cytochrome C, that might be the marker you used here, but
Yes, that will be.
it may not always reflect what's going on with mitochondria.
Yeah.
Right?
The goal will be also to use different probes that can assess different.
That's right. So
yeah. We just have to find the right ones, the right combinations for detailed study.
So some of the feedback we'd like from you guys with respect to the CYTOO is how would you go about validating something like this besides what we've done, and the idea of going through many more compounds, both negatives and positives, because even negatives would have an effect on some parameter but not be constant. Right?
That was my main worry, is that these very, very, very small changes with your false negatives.
Yeah.
difficult to say that until you have a really good database with a negative.
Well, all you know is the false negative has a change. You don't know what that change means yet.
But it's so small, it could be
Well,
meaningless.
and I'm trying, so the way we have picked those false negatives, let's be careful about this one, on that 100-time max, for example, lidocaine is one of those. Lidocaine has a beautiful effect on mitochondria if you go high enough. You can even trigger cell toxicity, but it's really high. So I think when I'm looking at Xi'an, when we defined those false negatives, a lot of this was either not detected in the high content we had done, I think, right?
Yeah.
And some of it was on that 100 times Cmax type. The lidocaine should have actually been declared as a negative component, anyway, not as a false negative component. So, I mean, that's the thing, right? So, from a toxicity point of view, you can measure whatever, you can make it either, more sensitive, less sensitive, I mean, we know all of this. And you get to a certain predictability and you work up your data a certain way, either, a rank ordering or, an exposure normalization type process. So I think that was, where some of our, question came from. So how much is that applicable against, a cytotox assay we run anyway? I think a lot of it was really trying to understand the biology piece, right? So now you have two patterns, and one time the mitochondria is all around the nucleus, and maybe that's a model for looking at X, right, versus the other time they don't even build a reticulum, so that's a model for Y. And I think that's kind of some of the work that I've seen, that I find that very intriguing. The other thing too is, can you in your system, so I can do whatever I want, and Padma can speak to that. I always have a 20% error to start out with. Any kind of membrane potential, is 20-30%. So one of the questions we asked here, where's the micropattern? Are we getting away from that? And you had not finished that validation of saying, yes, I can do this again and again and again. Because, that would be some of the nuances maybe when you look again back into biology that you need to pick up to make a statement. Because this is way too precious and labor intense, to look at dead or no dead, right? Yeah. So, some of the recent publication that came out around, the mutations and looking at, how under different conditions the fibroblasts would look like, or, what does media or culture conditions do to that, So I think those are the questions, you really still answer. I wouldn't worry so much about, predictivity. That's just the way we look at data. But can you have nuances? Can you see nuances reproducible? Can you see things that, can you see something else about mitochondria that I can't?
On the
right?
Chevrolet paper,
yeah, and I answered it. So, can you see something about lysosomes that Xi'an has never even heard about?
And that's something that we, I'm sure, could do with this type of tool. It's just that the question would be, what would be the right cell line? What would be the right pattern?
I don't think it matters. I think that's almost something that you got to try out. You have to ask a question, you have to figure it out.
Well, but that is, so develop the question. So the question, and then the cell line, and then we can figure out the markers to go ahead and play with it. The other thing isn't just the pattern, it's also what is the pattern coding. Because the pattern coding isn't blind to the cell. It has something to do with.
So can every cell adhere to every pattern? No, right? No. So how is it that I have G2 like a certain pattern and the ELA cell like a pattern? Do you know?
It's not that they don't like. So a lot of cells will attach to multiple different patterns.
Yeah.
Maybe different size patterns is really one of the big differentiators.
Okay, so the size is more important.
The size is important. There are some patterns that are a little bit easier to adapt, or where the cells can spread better than others. They are slightly different, like the Y would be a perfect.
Yeah, so when you think about your academic partners, right, so I'm thinking 30 publications. Some of it was probably developmental, even trying to measure some of this. But, when people are intrigued by this, that we're not toxicologists, what do they want to do? More detail, or more reproducible, or more
more phenotypic,
No,
more in
Yeah,
vivo-like.
Yeah.
Because the other thing you can do with the patterns is actually make the cell behave in a more in vivo-like way. The one example that Sebastian gave is the cilia.
Yeah.
Cilia formation. I showed you some of the stuff with the neurons. We can get myelination of neurons on the patterns. The other thing that can be done is cardiac myocytes. The cardiac myocyte, iPS cardiac myocytes appear to have more of an adult phenotype on the patterns than they do the fetal phenotype. So, pardon?
Will you base your more adult-like?
This is by word of mouth by a farmer who wouldn't tell me more than that.
So
And it was more of a
structure of how they align.
It was the ion channel. He's an ion channel guy, so it was ion channel physiology. So looking at the ion channel pattern. Yeah. And we see also there's publications that show that with how they align, that you get more expression of certain genes versus other genes. But this is the one thing that I was really asking about was the fetal versus the adult. And we got him to admit that they're behaving more adult-like with more of the ion channel pattern expression. So that's something that we'd like to follow up on because we think that's really important.
So perhaps, to sort of get back to your original question, how would you validate it, or how would you?
What would you want to do with it too? Yeah.
Yeah.
I think that's why we were in the beginning talking about FG2s, because a lot of us had done the high content and we were wondering if you could, boost that outcome.
We now can.
But I don't know, that was 2 years ago that.
It's still important.
I don't know, is it?
I don't know. That's the right question.
So I think the point I was trying to make was that what we really struggled with as a project team, frankly, I think this data continues to show the interest of the possibilities, but what, by definition, a validation study, or at least maybe as we define it, you have to have comparator data. In order to validate something, you have to compare it to something. And what we really struggled with, what is your comparator data? What are you validating against? What is your reference standard? What is your gold standard? And we got it all wrong last time, or at least we didn't really leverage anything. And I'm looking at this and still wondering, what is your gold standard? What are you validating against? Because you have your 4-hour incubation. You have a lot of differences, and it's really hard. It's almost like you're saying, or at least to me, like you're saying, it's basically like a different offshoot. It doesn't really compare to anything we're routinely doing now, so there is nothing to validate.
Well, yeah, I mean, on that note, right, I mean, what was the goal? To increase the productivity, the overall productivity of our false negative space? I think that's where we set out, right?
Yeah.
But that data set that we gave you, it doesn't answer that, right? Because if you look back at the data set, which was slide 2 or 3, I think, that's not what we gave you. We gave you actin, we gave you the cortisol compounds. So we didn't give you, a whole bunch of toxic versus non-toxic, and then let you set your boundaries of how much small do you have to, to set your 10% false negative. But I think that's just not where your opportunity lies. The opportunity is to have more detail of something that we are only marginally or globally touch on, right?
So if we could do something like this with a very valuable cell that was, like, for example, a podocyte, to be able to look at renal toxicity of some type, would that be of value?
But it's the same thing. So if you're saying you're positioning this as a safety assessment tool, you've got to have the productivity piece, which is nothing but statistics of, the large compounds. That's the problem.
Well, I understand the large compounds.
So you don't want to do that, right? That's not what you're set up to do. So I think what you need to say is, whatever people measure, I can measure this better. So let's go to Silumen, because that is the other big platform that you guys were they go and they analyze the well. They have fields and they analyze intensity or, responders or whatever, right? And they actually started working on it at some point right before the acquisition by Predator and Simplification on doing, single cell in there, because they found that then, hey, they had populations, and maybe the populations were telling them the story, right? So then there's Primer with the flow, who can do a sequence of events, right, real easily. And then there's ITDD, who looked at much more of mitochondrial type of movement and morphology and all of that. So, like Ernie says, it's like, what are you trying to do, and then with, who's the platform that you're trying to show that you have more depth or more, So I think that's why the paper that just came out looking at, using yeah. Or I also agree, I had seen some of the pictures in the past with the neurons that you had shown. So again, maybe it's an easier way to do the 3D, because the 3D is not quite there with the imaging at this point, or the compound penetration at this point, or the oxygen gradient at this point, right? So maybe the 3D has more problem of penetration measurements that you have, and you can bridge that piece. Is that what you want to do?
One of the things that Yvonne is referring to is that we have the ability to make clonal cell acyclic. So essentially from plating cells onto a micropattern, one cell per micropattern, we can basically make the cell, multiple cells all in the same well, form a sphere. And a hollow lumen, they all look the same, all the different spheres of the well look the same. And this is right now with NTCK cells, but we think we can do it with other cell types as well. And the idea would be that maybe this could be something in between a 3D spheroid. And because it's a little smaller, and like I said, we don't have oxygen penetration problems, you don't have, we can image it, there's a lot of things you could do with that, and maybe it would have more predictive value of oncology for oncology targets, and whether they're really toxic, because the 2D surfaces don't, yeah. So that may be something that we are working on that.
So I think, the predictivity piece is difficult, right? Because it doesn't matter what after you take or after combination, you're getting stuck somewhere at a certain percentage. And you can fill and flux and increase the 5% here, 5% there, So you can take hepatocytes and say 50% predictivity, and then do cytokine or BSEC or whatever. You can keep going. And every time you do that, you lose some compounds over here and gain some over here, right? Mm-hmm. So I think, and again, if you want to make it a statistical piece, it's a large number of statistical backwards. It's kind of a simple exercise. So that's why 3D has stalled, right? Because nobody, not even Chuck, has the money to run 350 compounds steroids, right? So, but, there's many, many scientific questions that have very little to do with predictivity, but looking at phenotypes, the right culture conditions. The other day I was reading a parasites and I thought, don't the parasites make glucose? Traditionally, but we all culture them, the same. I mean, as you said, we wanted to be more in vivo-like. So then I would continue along that road and say, this is more in vivo because it beats. This is more in vivo because it has biochemical. This is more, and I get more consistent imaging, therefore my data is tighter, my data is earlier, I have more nuances, the sensors in.
But we do that as well.
Yeah, that's what I said. You don't need to, keep hammering on the toxicology where we're not biting, we're not taking it. We're not, like, we have shown more interest, all of us, I think, have shown more interest in your biology.
In the biology than in using that to detect toxics.
Yeah.
But still,
why would would you personally want to do that? Why would you try to utilize this to make a better assay than
because
the Lumen platform? Here you go.
because in a way, I can see that this is not easy technology, but if you look in the details at the results that were generated, for example, you can totally relate the results that you get from siRNA, knocking down a gene, with the molecules that are known to inhibit the same gene, and you get exactly the same profiles across several markers. And this idea we came to develop as if we have the right number of markers, then each compound will have a given profile, right? And if we profile non-compound with as many as we can, so that we have high resolution, every new compound will find its place in the landscape and be classified. And in this way, then we reach predictivity, which is statistical way to reach predictivity. And those are ways to reach predictivity, to work on the biology, to get models that are more physiological, bring you closer to predictivity. But those are two approaches. One is statistical, one is the biology. But the statistical approach is appealing because it's very robust in cell biology that is lacking robustness in analysis. It's very quantitative ways to look at them. And we didn't show all the features of that, but you take two maps and you subtract them, and you get the difference. It's visual. You can tell this. You see what I mean? So you can even touch for the lysosomes, for example. You can see that there is difference between this and this. You can subtract the two images. You know exactly.
I guess we just don't understand the translation to what that information is telling at this moment, right? So
Actually, the thing that seems difficult to me is you're seeing molecules that don't have a lot of toxicity having statistical effects at very, very low concentrations. And how do you interpret that when you're in totally unknown space? Where do you cut things off? I mean, things end up being very arbitrary where you cut them off. And, the thing we always deal with in toxicology anytime we're doing anything early is we don't want to throw the baby out with the bathwater and these kind of things. So the level of certainty in an early assay to make this for decision making, and it's really weighted evidence usually. You're not going to be making no-go decisions on this. But the thing that this is the first. I wasn't involved in the first one. I think, Pat, were you?
Yes.
But to me, where you set that line can be very arbitrary. And how do you decide where to set that line on any one of these assays?
You only would know that from a large retrospective study.
That's exactly right. You have to run enough numbers.
For historical notes, Young and Padma basically set the library. I do think there's a point to be made relative to this question, and it was actually Saluman, the first one that brought it to our attention. And as it turns out, now that they're out of business, it was Bain Capital that actually did the assessment when they were thinking about investing in Saluman. And the question comes up, is this a research tool to be used in discovery, or is it a safety tool to be used across the portfolio? And if you do the math, safety tools turn out to be financially much more lucrative because it's done on the entire portfolio, every compound that comes through. So if you're developing a business, it makes perfect sense to look for the safety markets. The trouble with that analogy is, and Bain Capital pointed out, and Saluman proved the case, and you brought it up a dozen times yourself, whether you've seen the assessment or not, you intrinsically understood, the hump to get over is much higher. To develop into the research community, they're used to dealing with new tools that are only marginally validated, if at all, and jump at these kind of new opportunities without a lot of investment. But if you're in the safety space, you've got this huge nut to crack called validation, which can be hundreds or thousands of compounds before people have the confidence that it's actually a predictive tool. And so you got this big expense with a high reward, and so
Possible high reward.
I understand why people sort of chasing that pot of gold at the end of the rainbow, because potentially it is very lucrative. But I keep coming back to the point I made first. In order to get there, you got to have comparator data. You got to show that it's as good at or better than how you're already doing it. And in order to convince communities of that, particularly the safety community and your traditional toxicologists, it's got to be robust, shall we say, which is a big thing. And I'm just repeating
your business.
Wondering, well, actually, we've done several things where the CHAN-TEST project, which you are familiar with, we just did, was exactly that. We had good comparative data, and we had their new test, and you could actually say, on a very statistical basis, to use your word, when was it better and when was it not better than the existing property. So there are a lot of examples out there where you can come up with a null hypothesis that it's no better, and then do the test to see if it is better than the existing way. But what is that null hypothesis in this case? What are you going to measure it against? You did say now you can do hefty details, because 80% of the liver data is FG2. If you're going to try to just compare to the literature as your reference standard, it's almost FG2. That's where it's at.
So I like what you said, right? I like the biology, but I'm trying to make it a little bit more realistic than what you're having on your dish right now. So, if you will take H92 cells to the amount of hormone-related hypertrophy response that they have lost. You have G2s, you have a beautiful blue-gal response, but you can't measure dihydroxysteroid. Can you do it now? So, it's more survey of what people say about the limitation of that particular cell line, that you can prove that, you have manipulated it to a way where you think it's, a good situation because you're not relying on primary cells. Or the other thing that I find very intriguing is, can you manipulate the technology in a certain way? Can you make one that has, no good lysosomal localization, or, it doesn't have a good filament. Those models look for questions.
Are those toxicology questions or biology questions?
No, I'm a chemist. So, it's all biology. So I personally, for toxicology, even if they would give me everything for free right now and say, Sebastian's come to live with you for 6 months, I would not have a toxicology question, but I would have 5 biology.
Funny point, because it makes me think of the Pellman paper, where you have cancer cells that could give multipolar divisions, and they would die. There is 3 axes of division, or division. They die. Or they don't divide, but they die. But in nature, these multiples, they get together. And usually these cells, they divide. And they survive. And they are cancerous. And they can. And Pellman used our technology, the guy at Harvard, and put the cells on Y-shaped micropatterns. So now he's forcing the cells that will give multipolar division to do 3-axis division, and then they die, of course. But he needed to do that to study those cells. Otherwise, they do
So that's what I'm saying. If Sebastian is coming to see me for 6 months, they'll have a lot of biology stuff we're going to know, but there won't be a survey.
Wow, you have a lot of money. You won't Sebastian for 6 months.
I'm just trying to explain where I'm at.
I'm expensive.
My daughter, I just don't, I would not be able to come up with a meaningful experiment with
I've got another meeting that started at 8:30 that I've got to be at, and I'm very interested, and I'm embarrassed for leaving, but I've got to leave.
Thank you.
Patrick's here,
Thank you.
I haven't seen him.
So just out of curiosity, for the CYTOO group, are there, just maybe to throw it back and flip it the other way, are there things that you guys have thought of that would be safety applications or other people have told you about, or toxicology, and is there anything that...
I think the safety is still, trying to make it more, realistic. So if I have to choose, it does have from the title side. So all of those is all good. All of those is good. But I would stay away from that 300 compound predictivity piece. You're going to fail.
We also are thinking of FG2 cells.
Yeah.
Or primary liver cells, yeah.
I don't know. I mean, FG2, but you really do FG2. I mean, they have these new RSLs or whatever. They have some better cells. Why would you go back there?
So the question is the FG2 cell a bad cell because of the way we grow it?
So yes, and I agree.
that's
You know what, I
actually-
just brought it up. So did you know that if you run them to confluency, they have perfectly fine 3A4? Even though everybody says they have no 3A4. They do. If they could go confluent, they have the
Cells
same
behave
3A4.
really differently depending upon the culture medium you have them in and the growth.
Glugel, isn't that the craziest thing? Like, we run a glugel in there, that looks beautiful, right? But we can't measure dihydroethylene. I mean, how can that be? So I think there's a lot of opportunities, and you have started that anyway.
They're primarily erythroid in nature.
Pardon?
They're primarily erythroid cells.
Erythroid.
Mature cells that secrete EPO in the liver.
Oh,
So, yeah.
I didn't know that.
Yeah, they do. And maybe Yvonne was mentioning that they have make cell-cell communication, they mature, and then they have all the 3A4s they want to do. Could be. But, as Bernie was saying, you could adopt another cell. I mean, if the question is, do you want to see mature liver components in there? Is that what the question is? Or are you trying to predict mitochondrial toxicity or organelle toxicity, or are you trying to
I think the first thing would be is, can you make the cell behave more like a primary in vivo like cell? And once you get there, then you can go to the other things.
Right.
The first thing is, can you make that cell?
And so you haven't shown, the greatest validation data from your intra, you say 2 out of 3, which is already again. But looking at the images that I have seen, I think you can really pick up nuances. We don't see nuances with mitotracker or TMRM, where, we have to see nuances. So compartmentalization and the nuances, I think is very intriguing. So for example, if I have 5 different compounds that affect mitochondria, are they behaving differently and how they change? So again, comes back to biology. Can we understand it? Can we target it, right? So I have, mutants of in old, Yvonne fibroblasts and young Patra fibroblasts, and we're trying to look at response and what is that, sensitivity of you picking that up. And I think you can perceive images. How good you are to repeat that from Monday to Tuesday, that's something you guys got to work on, But I think
I couldn't have the data for today.
biology,
Tomorrow.
I think you're good at repeating the biology. I doubt if you see today transportation this way, tomorrow it will go that way, right? So I think you're really good at that. If you have to make numbers out of it, that's a different question, how good that looks from day 1 to 2, But I think you have so much sensitivity and opportunity there for really taking something under a lens, right, a magnifying lens. And so the only other ones I've ever seen trying to take this approach is really ICDD, which I guess you guys are familiar with, you know, where they looked at 14 different ways to look at mitochondrial movement. Was the same thing, you know, they were trying to figure out how predictive this was of toxicity rather than saying, how does it look in pink-bargain interactions?
Clinical toxicity.
Yeah. So, but you have so much, you know, a potential, and you have already shown a lot of that. So, I mean, a lot of people are already looking at it, you know, in the right way. You have a lot of customers you showed that have started to look at it in a certain way for their biology, right?
Even for simple things, like getting the cells to flatten out so you can do siRNA transfection. I mean, it's super
This
simple.
we have data on.
I know
And
we've
what
done
is
it,
your
but our
business model at the end anyway? Is it selling, you know, your software packages, is selling the pattern plays, you know, because
and selling services, and not necessarily that we would do things, but selling like assay development with either the company doing the assay development or us working with the you know, the farmer or the partner to do the assay development on new projects, you know, to try to validate. And so those are some of the things that are ongoing.
Yeah.
I think this is really cool. I think it's just not
really
predictive.
cool.
I don't want to use it.
No, that's, when you started showing the data, we went back to the ICDD data that we saw. It's kind of really cool that you have this patterning around the effect of a compound. And I was thinking, adding to what Yvonne was saying, is you have 5 different mitochondrial toxins, for example, but they have their own signatures. And to me, it's more like you have one already. Now it's more building around that. Maybe it's a fluorescence intensity measurement of a functional dye, or maybe it could be a biogenesis type marker, who knows. But I mean, it's kind of bringing all together and kind of layering over this pattern. More multi-parametric way I'm thinking about it.
And again, the pattern must be more sensitive than, you know, for example, everybody doesn't run a high content. They have pattern recognition too, where you do heat maps, you know. So these all show an increase in ROS first that drops membrane potential and affects glutathione. So they can do this too, you know, the heat map generation and the similarity patterns. But here, you're looking one beyond that, right?
Yeah.
So you're not quite on, you know, the protein interaction level, but you're just one above from, you know, you're in between the overall simple readout and more delineation. So that's, you know, looking at themselves and how they are more real and looking for really changes hypothesis driven.
So let me ask a question. You guys weren't part of this conversation before, but one of the things we started to do in the Technology Evaluation Consortium is offer up an opportunity for the company after a normal summit, which is this is the second summit, for you guys to present to a wider audience back at the pharmaceutical companies. So I guess I'd like to ask you guys in the pharmaceutical companies who are here, do you think that this is provocative or interesting or informative? Because I'm hearing that it's not definitive, and you know it's evolving and it's learning and it's growing and you know it's looking for applications, et cetera, et cetera. Do you think that it's at a place where it would be interesting to have a wider exposure to what they've showed to your companies? So maybe we'll start with Stella.
I have interest in this technology, but I want more data or evidence about the distribution of organelles and toxicology. So I think some parts
And you're thinking even more broadly beyond the toxicology group, would more people in the company be interested in hearing about what they have today?
I think so. I will tell my boss to this is interesting and true.
And you haven't seen,
We could share much more.
you haven't seen the data on the neurons that we
Yeah.
have seen, or cardiomyocyte current, you haven't seen it.
There's a lot more that has been done. This is just a tiny, tiny part.
Yep.
Focus mostly on this particular tool.
So on that note too, so Pfizer's a large organization, I was unaware of that. Cambridge was actually doing some work on, and they should, everybody should, but I think what it also would allow is, to start these conversations, because they might be going down the road and say, oh, that doesn't work, because they're just all by themselves and they're not asking, right? And so,
Cambridge of Pfizer.
yeah,
Or the Cambridge
so,
office of Pfizer.
I think that would be something to keep in mind too, actually exposing this to other parts of the organization to make sure that there's a good communication scheme. So, not that they try something that, it wasn't meant to be, and then there's the big disappointment.
Yeah.
Right.
So I would be very, now after all we have been through, if somebody in Cambridge toxicology would say, oh, I heard about CYTOO, I'm going to use it for hepatotox, help would put on the brakes really fast, versus CNS would be different, So, from your list earlier, you are already working with quite a few people, but you might not even be aware of that.
How about for Amgen? What are your thoughts?
Well, when I first started seeing those slides, the first slides which saw the questions that popped up regarding transporters. That's a big field we are involved in. A lot of applications on the drug metabolic arena. And just going into that a little bit deeper is, if you're into, you have the technology to do all these patterns, so why wouldn't you do I'm just describing it loosely, as a structure of a liver, with all the hepatocytes and stellate cells and all those right things. I'm just asking
No, that's a good question. No, that's
visual thinking. But what I'm saying is, you could really understand.
To build a liver.
Yeah,
that's part of a project, of an R&D project, actually.
So that's one application. And going back to, again, drilling down into the organelle toxicity, it's intriguing that you have a great anchor there with the patterns. And now for me, it would be building, we see all sorts of organelle toxicity in the portfolio. So my question there are many ways of looking at it. For example, lysosome LAMP1, those kind of things. And there's the lysosomal pH part. So it's a whole, variety of things that you need to add to this anchor that you have created. So that's how I'm thinking about this.
Okay, great. How about Pfizer? The west coast of Pfizer.
Yeah, personally, I think it's very, very interesting. I talked to Lisa quite a bit before this. I think, from my sort of personal interest, some perspective, I think we can get the answer, get some question answered that we couldn't use the regular 2D. Although the technology is not mature, but I think it's okay, I guess, to share with, because people have probably a different angle. The platform is here. All they need is to find whatever the endpoint that works for whatever the question they have. So I think it's good enough to share.
How about for Mark?
Top aspect, I agree with Audrey, the discussion here is not really much proof yet for me to switch from a standard model, and as Ernie also said, what is the comparator here? And 2 years ago we said FG2, and I think the last 2 years that's also probably changed. Everyone's working on 3D models and growth culture models. I do think it's a great idea to take old cells like FG2s and to prove that you can still use them. They have hepatic function. And a lot of guys in projects I'm working on outside of work that are really pushing FG2 cells all the time. because they say they have models where they can really improve the metabolic competency and the hepatic functioning. And if you go down that way, it would maybe highlight a bit more for toxicologists. But I see no reason why other guys, there's a lot of self-culture guys at Merck, seems everywhere.
Yeah.
Maybe not quite so in depth as the bigger companies, but we definitely have people who will be interested to have all sorts of subsystems that they want to implement for whatever reason, pharmacology. If the cells really behave better and more sensitively on these micropatterns later, why shouldn't they? And I presume you've never had contact with our...
No, that's not been our traditional focus, but we're trying to branch out.
That everybody
I think the talks, especially at Merck, everything's been cut so much, we're not even doing any what high content imaging system, and we don't use it since a year because it was. So I think.
She'll put it right outside her office.
That would wake them up, maybe. Oh, I'm shipping a lot of spiders.
It's sort of intrinsic. The idea of stacking the images up and creating an image because they're so reproducible with their organization and stuff. It just seems like that ought to be self-obvious that it's valuable.
Yeah.
It just looks like something that you should be able to leverage. That's a really nice idea. You have enough data to show that it in fact works. But now you've got a really nice idea that's not really a great application. Exactly. And maybe you have in the discovery, but I mean, for what we're talking about, or what this is about, safety assessment, how are you going to take this idea and translate it into, I'm all about prediction. I know that you think it's probably not there, but where do you, because this seems like it's tailor-made for prediction, increasing reproducibility, increasing or decreasing the variance